What's up, TUMNAD Lectures? In this video, we are going to be looking at what we call the intestinal villi. Let's go ahead and get started. When we talk about the intestinal villi, these are going to be like finger-like projections which are going to be residing within the mucosa of what we call the small intestine. Very short structures that are primarily going to be increasing the surface area for what we call absorption of different nutrients. So let's go ahead and see the anatomy of all those structures that really make what we call. Now, when you see this kind of the villi, of course, when we talk of one, it's going to be now the villus. But when they are like two or more, it becomes what we call the villi. Okay. So now, when you see this villi, we're going to be looking at the villus anatomy. We look at the enterocyte. We also look at what we call the brashborder enzymes. Okay. Now, when you look at the whole of this structure, the whole of this one, only this one is what we call the villus. The moment you have more than one, it becomes what we call the villi. Okay. Now, looking at these structures down here, you see these are going to be one with blood vessels, okay? Blood vessels and even some part of what we call the lymphatics. Now, this one, which is a bit blue, this one is what we call the venue, okay? This is what we call the venue. And then this one, which is green, this one is going to be what we call the lymph vessel, okay? This is what we call the lymph vessel. And then this one here, this one is going to be now an arterial. Okay, this one is going to be what we call an arterial. Okay. Then as we move up, we have this network of the blood vessels. Okay, this one here is simply called network of blood capillaries. Okay, it's simply called the network of blood capillaries. Okay, network of blood capillaries. Okay. So now when we come to this center this tube is super very important in absorption of what we call the fats. You know how it is called? This center the tube, this is what we call the lacteal. Okay, is what we call the lacteal. Super very important in absorption of what we call the fats in form of what we call chylomicrons. Okay. Now, when you look on the peripherals of this villus, you're going to be seeing these epithelial cells. Okay, these one here are going to be what we call the epithelial cells. Okay, what we call the epithelial cells. Now, and of course, this epithelial cell is going to be having these tiny of the extensions which you call the microvilli. So now, we are going now to be zooming on one of the epithelial cell. We zoom in out and see exactly what are some of the structures that are going to be contained in what we call this the epithelial cell. Now, this epithelial cell is going to be one of what we call the absorptive cell of what we call within the small intestine. Okay, and another name for the epithelial cell is what we call the enterocyte. Okay, so now this structure here, the whole of this cell we have just extracted here and we have zoomed in. Now this one here is what we call the enterocyte. Okay, what we call the enterocyte. Now we have zoomed it out and then we want to see what is exactly going to be contained within the enterocyte. Now, when you see the whole of these the extensions, the extensions on what you call the apical side, okay, or the apical side, you have this the extension which are like the bristles. You know how these extensions are called? These extensions are going to be the one which are going to be on all of these these enterocytes. Those extensions are simply called the microvilli. Okay, these ones here are what we call the microvilli. These the microvilli are super very short. They are 1,000 times shorter than what we call the villi. Okay, super very short. Okay, now within this this microvilli, within this the microvilli on the apical side of what we call the enterocyte, we have a couple of what we call enzymes. Okay, now this microvilli that makes this of the brush. Okay, this shape is what we call the brush border, just because like the bristles on what we call the brush. So now these are making what we call the brush border, and because they are going to be containing a lot of different enzymes, these enzymes we just simply call them the brush border enzymes. Okay, we are going to be having tons and tons of different enzymes that are going to be breaking down the food chemically into an absorptive form. So now let's go ahead and see what are some of those examples of enzymes that are part of what we call brush border enzymes. Now, for those the enzymes that are going to be dealing with anything concerning carbohydrates, okay, they are going to be what we call the saccharide, peptidases. Okay, the examples of these ones, we are going to be having what we call the lactase. Okay, we have what we call the lactase enzyme that's going to be breaking lactose, and then we also have what we call maltase. Okay, we also have what we call the maltase, and then of course the maltase can also be called what we call glucoamylase. Okay, it's going to be dealing with the breakdown of what we call maltose. Then we also have what we call sucrase. Okay, we also have what we call sucrase. Super very important in the breakdown of what we call sucrose. And then the last one is what we call dextrinase. Okay, what we call dextrinase. Super very important in breaking what we call the alpha limit dextrins. Okay, and of course dextrinase can also be called isomaltase. Okay, this one can also be called isomaltase. Okay, isomaltase. So if you don't want to call it dextrinase, you can even call it isomaltase. Super very important in breaking in breaking one of the beautiful kind of the bond which is called alpha 1,6 glycosidic bonds. Super very important in what we call the breakdown of alpha 1,6 glycosidic bond. Then when we come to the proteins, some of those kind of the brashborder enzymes that really deals with what we call proteins. The first one is what we call dipeptidase. Okay, dipeptidase, and then another one is super very important is what we call aminopeptidase. Okay, aminopeptidase. So now these are some of the key most important enzymes which are part of the brashborder enzymes dealing with what we call nutrients, which is going to be in what we call the proteins. Okay, there are even more others like enteropeptidases. Super very important, making sure that we have what we call chemical digestion within the small intestine. Okay. So now this briefly covers the intestinal villi. Let's just have a very quick recap. We have said that this one here are going to be what you call the venules. We're going to be having what you call the lymph vessels, the green ones. We're going to be having what you call the arterioles, okay? And then also have what you call a network of blood capillaries. This one, a network of blood capillaries. Then we have this central tube which you call the lacteal that absorbs what you call fats in form of chylomicrons. Then we also have what we call the epithelial cells, which you call the enterocyte. When we zoomed this kind of the enterocyte, we said that it's going to be having what you call these finger-like tiny extensions that are 1,000 times shorter than the villi, which you call the microvilli, that creates that kind of the brush border. It's going to be containing a lot of enzymes. A lot of enzymes will have what we call those ones which are dealing with carbohydrates. We have what you call the lactases, the maltases, the sucrases, the dextrinases that are going to make sure that the nutrients are being broken down into that final kind of absorption format. Then also have those ones dealing with the proteins, which you call the dipeptidases, and even also what we call the aminopeptidases. Okay. So now, TUMNAD, this briefly covers the intestinal villi. If you think whatever I've talked is making sense, is adding to you, we are benefiting, please can support me. And that one can be done by a couple of means. You like the video, comment down in the comment section and subscribe. Thank you very much for watching. TUMNAD as always. Until we meet next time.